the next pathway which we are going to see today is hexose monophosphate pathway. It is often referred to as HMP in short, or this pathway do also have certain other terms, or it is also known with other names also, like your hexose monophosphate shunt. In short, it referred to as HMS, or pentose phosphate pathway, or PPP, or phosphogluconate pathway, PGP, or based on the name of the biochemists who have described this pathway for the first time, Warburg and Dickens pathway, W&D pathway. Uh, so all these names, be it hexose monophosphate pathway, be it your hexose monophosphate shunt, pentose phosphate pathway, your phosphogluconate pathway, or the Warburg and Dickens pathway. All these are one and the same things. Now, what is the function of this particular pathway? It is used in fermenting various different kinds of carbohydrates. Now, often it is referred to as a shunt or a loop in your basic Emden-Meyerhof-Parnas pathway. Now, what do you mean by a shunt? You may say it is a bypass or a shortened route adopted by microorganisms for fermentation of carbohydrates. Why do microorganisms do this? Why do they adopt a shortcut, or why do they take a loop to EMP pathway? We'll see later on. Now, what basically happens in this pathway? Unlike your EMP pathway, HMP pathway yields only half the energy. So basic function of HMP pathway is not to yield the energy. The function of this pathway is that it provides for the ribose phosphate required for nucleotide synthesis. And secondly, it provides for the reducing power in the form of NADPH2. So that will be the basic difference between your EMP and HMP pathway. You should always remember this thing, that for EMP also and for HMP also, the glucose enters into the pathway, but EMP pathway brings about a breakdown of glucose molecule to yield the pyruvic acid and results in generation of about 8 molecules of ATPs. Whereas in HMP pathway, energy is not produced in that amount as that of your EMP pathway, but it results in formation of one of the raw materials of biosynthesis, that is ribose phosphate. Now, this ribose phosphate is one of the very prime requirements needed for synthesis of nucleotides for making the RNA molecule. And similarly, it may be later on utilized for making the DNA molecule also, if it is converted to deoxyribose. So, HMP pathway is basically providing for raw material of nucleotide synthesis. Secondly, it results in generation of NADPH2 molecules. Now, these NADPH2 molecules are acting as the reducing power during various different pathways later on. बाद में जब कोई pathway run होता है, biosynthesis होती है, so during that biosynthesis or those pathways, these NADPH2s will be utilized as the reducing powers. Okay. Secondly, furthermore, the pentoses may be oxidatively degraded to hexoses. which later on may enter the glycolysis. Means this pathway does not functions from one side to another only. Means usually if you see the EMP pathway, a 6 carbon compound, a hexose sugar enters into the pathway and results in breakdown of the glucose, and the glucose is exhausted at that. Whereas this pathway functions in both ways, means hexose will be broken down to convert it to pentose sugar ribose, and vice versa may happen. The pentose sugar may be converted to form the hexose sugar, which may be utilized for the purpose of glycolysis. Say, if cell is not in need of ribose sugar, cell is starving. There are two conditions. When cell is present in a natural environment, there are two possible conditions: that cell is getting the ambient nutrients, supportive growth environment, and all those things. In that case, cell will go for proliferation. It will try to multiply, it will try to increase its population. For that purpose, it requires biosynthesis. Means new molecules should be synthesized so that a new cell could be made. Okay. But if cell is under starvation, it is not getting proper nutrients. So in that case, first necessity of the cell will be that it should sustain itself. If cell doesn't die, then it could multiply. If there are no nutrients, if there is no source of energy, then what is the use of replication? What is the use of making new molecules of DNA and RNA and all that? So in such cases, the primary objective of the cell would be to synthesize the energy required to sustain the cell first. Okay, so in such cases, this HMP pathway may convert the pentoses to hexoses, because pentose cannot enter glycolysis. So HMP pathway converts these pentoses to hexoses, and these hexoses then enter into the HMP pathway. Okay, so that will be the significance of this pathway. Now we'll move on to the actual reactions, how they take place during the pathway. So as for any pathway, the entry molecule is your glucose 6-phosphate. Basically, it will be glucose, D-glucose. Okay, the glucose will be acted upon by hexokinase enzyme and it will be phosphorylated to form this glucose 6-phosphate. Okay, it is the phosphorylated form or charged form of your glucose molecule. Okay, now it has entered the pathway. The enzyme glucose 6-phosphate dehydrogenase acts upon glucose 6-phosphate and brings about dehydrogenation of the molecule. Okay, this glucose 6-phosphate will be dehydrogenated. Now, what do you mean by dehydrogenation? Dehydrogenation means removal of hydrogen from the substrate molecule. So means glucose 6-phosphate dehydrogenase will be bringing about dehydrogenation of this glucose 6-phosphate molecule. Okay. So this enzyme, glucose 6-phosphate dehydrogenase, will bring about dehydrogenation. Hydrogen will be removed from this glucose 6-phosphate and it will be converted to 6-phosphoglucono 1,5-lactone. Okay, and during this process, as there is removal of hydrogen, so there will be a requirement of a hydrogen or electron acceptor molecule. So here, this NADP acts as the acceptor of this hydrogen and is converted to NADH. Okay, and this NADH is going to serve as the reducing power during the biosynthetic reaction. This is the first significant point of this particular pathway. As what we have said, that this pathway helps in generating the reducing power in the form of NADPH2s. So that's how the NADPH2s are generated. Glucose 6-phosphate is being converted to 6-phosphoglucono 5 delta lactone. Now, once this phosphoglucono delta lactone is formed, it is again acted upon by another enzyme called glucanolactonase, and this glucanolactonase converts this 6-phosphoglucano 1,5-lactone to 6-phosphogluconic acid or 6-phosphogluconate. Okay. This 6-phosphogluconate is again acted upon by another dehydrogenase enzyme. Now, similar to the first reaction, dehydrogenase enzyme again removes the hydrogen from the substrate. In first reaction, glucose 6-phosphate was the substrate. In this reaction, 6-phosphogluconate is the substrate. Hydrogen will be removed. Again, the acceptor of hydrogen is NADP. It will take up the hydrogen and get converted to NADPH2. Okay, and this 6-phosphogluconate is converted to ribulose 5-phosphate. Now, here you would see that this 6-phosphogluconate is still a 6-carbon compound, whereas it is now converted to ribulose 5-phosphate. Okay, then this ribulose 5-phosphate undergoes splitting. An enzyme called ribulose 5-phosphate 3 epimerase brings about splitting or breakdown of this ribulose to xylulose 5-phosphate and ribose 5-phosphate. Now, this ribose 5-phosphate is the second significant issue of this particular pathway. What we have said that this pathway is going to generate the raw material, the pentose sugar, ribose for the nucleotide synthesis, and here is that ribose generated. If the cell is in need of nucleotide synthesis, this ribose will be picked up from this point and will be utilized in the nucleotide synthesis. Okay. Now, the xylulose 5-phosphate, if suppose the ribose 5-phosphate is not utilized, what is going to happen? Cell is not in need of that nucleotide synthesis. What is going to happen then? Another enzyme, transketolase, will act upon this xylulose 5-phosphate and ribose 5-phosphate. A condensation and splitting reaction will happen. Transketolase, what it is going to do, it will take the substrate xylulose 5-phosphate and ribose 5-phosphate. First, it will condense both of the molecules. It will make a sugar molecule consisting of 10 carbon atoms. Okay, and it will then split both the molecules to a 3 carbon and a 7 carbon compound, pseudohaptolase 7-phosphate, and this glyceraldehyde 3-phosphate. Where from we are getting this? We are getting from xylulose 5-phosphate and ribose 5-phosphate. This is 5 carbon, this is 5 carbon. Both are condensed, make a 10 carbon compound, a 10 carbon sugar, which is then again splitted down to form pseudohaptolase 7-phosphate and glyceraldehyde 3-phosphate. Okay. Now, the significance of this glyceraldehyde 3-phosphate, you know very well. What this glyceraldehyde 3-phosphate is, if the cell is in need of energy, this may enter into the EMP pathway. This glyceraldehyde 3-phosphate it may enter into the EMP pathway and continue to form the ATPs. Okay. Again, an transaldolase enzyme acts upon this pseudohaptulose 7-phosphate and glyceraldehyde 3-phosphate if both of these glyceraldehyde 3-phosphate is not utilized. Okay. Now, one thing you should remember that the most of the reactions in this pathway are conditional. If suppose this glyceraldehyde 3-phosphate is consumed for EMP pathway or for any other biosynthetic reaction, this reaction will not take place. Means transaldolase will not act upon anything. The pathway will be blocked at this point only. Okay. The pathway will progress only when there is glyceraldehyde 3-phosphate not being utilized by any other pathway. Okay. So we just assume that there are pseudohaptulose 7-phosphate and glyceraldehyde 3-phosphate present in the cell. The transaldolase enzyme will act upon both of these, condense them, and then result in formation of one 4-carbon and one 6-carbon compound, erythrose 4-phosphate and fructose 6-phosphate. Okay. Now, what is this fructose 6-phosphate molecule? You know very well, fructose is the isomer of glucose. So if you are having fructose, that means the cell is having glucose molecule. So this fructose can enter the EMP pathway and then result in formation of the energy. Okay. Again, this xylulose 5-phosphate couples with this erythrose 4-phosphate here. If suppose this ribose is utilized, so xylulose will be remaining, and when this glyceraldehyde is utilized, now the question arises, just now I have told that if these products are not being utilized, means if ribose 5-phosphate is not utilized, the pathway is going to function in this manner, means this reaction transketolase will act upon ribose 5-phosphate and xylose 5-phosphate condensation and splitting. All these things are going to happen when ribose phosphate is not being utilized. And again, I have told that if glyceraldehyde 3-phosphate is not getting utilized, then transaltolase will act upon it and result in formation of erythrose 4-phosphate and fructose 6-phosphate. What if the cell utilizes ribose 5-phosphate? I have said that this reaction is not going to happen. Transketolase will not have the substrate and will not act upon anything. No pseudohaptolase or glyceraldehyde 3-phosphate is going to be formed. And if this thing is not being formed, further reactions will also be blocked. Means at this particular point, if ribose 5-phosphate has been utilized for biosynthesis, the cell will remain with this xylulose 5-phosphate. Okay, it is not going to be utilized or being acted upon by any other cell. Now, what happens? Another glucose molecule is used by the HMP pathway. Similar kind of reactions occur, and this xylulose 5-phosphate, again, one other molecule re-enters at this step. It combines with erythrose 4-phosphate and will continue the pathway. Okay. If you see or if you compare this thing with your EMP pathway is straightforward pathway. It is a cascade-like reaction. If first reaction occurs, second will happen then only. If first reaction is blocked, second will automatically be blocked. But in this pathway, why we are calling it a shunt? Because there are many alternate ways by which organism could metabolize the sugar. As we are seeing here, after utilization of ribose, eventually you may say that this xylulose 5-phosphate is now a byproduct, a byproduct. It is not going to be utilized by cell. But what cell is doing? It is taking up another glucose molecule, and when it forms this erythrose 4-phosphate, okay, दूसरा ग्लूकोज मॉलिक्यूल वो लेगा, उस ग्लूकोज से एक फ्रुक्टोज 6-फॉस्फेट यहाँ पर वो तैयार करेगा. ओके, नाउ दिस फ्रुक्टोज 6-phosphate may enter EMP pathway and can directly result in formation of energy. Okay, again in this case, this erythrose 4-phosphate would be a byproduct. It should be considered as a waste, but it is not wasted. How it is not wasted? Because we again have another thing which is not being utilized, xylulose 5-phosphate. Okay, so if you see this transketolase enzyme, what does it do? It takes up this xylulose 5-phosphate, takes up this erythrose 4-phosphate, condenses both the molecules. Now, if you see, this is a 5-carbon compound, this is a 4-carbon compound. 5 plus 4, you get a 9-carbon carbohydrate, and then this condensed mixture is broken down again to result in formation of another molecule of fructose 6-phosphate. This is a 6-carbon compound, and one more molecule of glyceraldehyde 3-phosphate. Now, here there is one fructose 6-phosphate, here there is one fructose 6-phosphate, here there is one glyceraldehyde 3-phosphate. All these three molecules, they may enter your EMP pathway quite easily, and they be utilized to form the ATPs. Okay. Now, this glyceraldehyde 3-phosphate, which is being formed here as a result of this transketolase enzyme, if suppose it is not being utilized, there is one more possibility that another enzyme called triphosphate isomerase may act upon glyceraldehyde 3-phosphate. If you remember your EMP pathway, in EMP pathway there is one splitting reaction. What happens? Fructose 1,6 biphosphate gets broken down into glyceraldehyde 3-phosphate and one more compound, that is dihydroxyacetone 3-phosphate. That 6-carbon sugar is broken down to 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone 3-phosphate. Okay, now this glyceraldehyde comes to this point. Here we are accumulating one glyceraldehyde 3-phosphate molecule from xylulose and erythrose. Okay, so this glyceraldehyde and this dihydroxyacetone phosphate, they are acted upon by transaldolase molecule, and eventually they result in formation of fructose 6-phosphate molecule. Okay, so if you see the overall pathway, you will get an idea why we are calling it as a shunt. हमने क्या कहा start में that this pathway is a shunt to EMP pathway. It's a bypass to EMP pathway. Now, if you compare the entire pathway with the EMP pathway, what you observe that EMP pathway is straightforward. One after other reaction occurs. If the product of one reaction is blocked, the subsequent reactions will automatically get blocked in EMP pathway. But here what you observe that even though the intermediates of this particular pathway are being utilized, like this ribose 5-phosphate is utilized, or say this glyceraldehyde 3-phosphate is utilized somewhere, or rest of the other molecules are being utilized, but this particular products of this pathway are being utilized by other biosynthetic pathways, then also the leftovers, the unutilized components of the sugar catabolism still could be reused through shunt mechanisms. Like you see here, this xylulose 5-phosphate recombines with the erythrose 4-phosphate arising from another molecule of glucose, okay, and will eventually lead to formation of this fructose 6-phosphate. Okay, this glyceraldehyde 3-phosphate may directly enter into the EMP pathway and will be converted to pyruvic acid. Okay, or if suppose both these glyceraldehyde 3-phosphate, this one is not entering into the EMP pathway, and again this one also is not entering into the EMP pathway. So what will happen? This glyceraldehyde 3-phosphate will be converted to dihydroxyacetone 3-phosphate, and again a condensation reaction will occur. It will lead to formation of fructose 6-phosphate. Okay, so 6 carbon compounds, this fructose 6-phosphate. Here we are seeing this one, and this one. What are these molecules? If these molecules are generated, they may enter the glycolytic pathway, EMP or ED, whatever the glycolytic pathway the cell prefers. These molecules will enter there and will be used for generation of energy. Okay, that's how we justify what we have said, that HMP pathway works in both the directions, which means in one way it will convert a hexose sugar to pentose sugars, and secondly the unutilized pentose sugars will be reconverted to a hexose sugar. And if you see, although in case of MP pathway, glucose is the entry product or entry substrate of your EMP pathway, but eventually what happens? Glucose is converted to glucose 6-phosphate, then it is converted to fructose 6-phosphate. Okay. Third reaction of your EMP pathway. First reaction is phosphorylation of glucose to glucose 6-phosphate, and then second reaction, glucose 6-phosphate to fructose 6-phosphate. So here we are getting that molecule directly, fructose 6-phosphate हमको यहाँ पर directly pre-made form में मिलता है. And if cell is in need of energy, this fructose 6-phosphate could be directly utilized for synthesizing the energy, similar to that of your previous one. EMP pathway में ये enter होगा और वहीं से सारी reactions फिर continue होगी. Or if any emergency need arises, this glyceraldehyde 3-phosphate may be utilized directly during this pathway. Okay. So this is how your HMP pathway functions. ये है आपका HMP pathway, hexose monophosphate pathway. There are other names also to this particular pathway. इस pathway के लिए दूसरे terms भी आपके पास है. Just you should remember all these terms in your examination. You may be asked with any of these terms. Don't get confused. Hexose monophosphate pathway, hexose monophosphate shunt, pentose phosphate pathway, phosphogluconate pathway, and WND pathway. All the 5 terms are the name for same pathway, HMP. Okay, the significant points of this pathway, that it results in generation of NADPH2, reducing power for biosynthetic reactions, and secondly, it results in formation of ribose sugars required for nucleotide biosynthesis. Okay, so this much for your HMP pathway. If anybody of you have any questions, you may kindly ask. Any questions or doubts regarding this?